Does it work? Yeah. I'm losing time, so. Yeah, thank you very much for the invitation. So what I will talk about is the carbon in nutrient cycling response to elevated CO2 fumigations, particularly at BIFoR FACE for the first 5 years. And that's the sink in the forest. And talking about the land carbon sink, if we look at the sources of CO2 emissions from human sources, they all end up in the atmosphere, but 1/3 of that is taken by the land system. And within the land, forests are a substantial sink for that human emitted CO2 in the atmosphere. This sink, the land carbon sink, is predicted to be enhanced under future climates where the CO2 would be higher. And if we look here, the land carbon response based on these publications, which is averaged by the blue bar, but the individual observations are results from different modeling studies showing a huge range in the predicted response of the land systems. And when they accounted for the nitrogen as a nutrient, as a limiting nutrient for photosynthesis, that response was pretty much curtailed by about 55%. So the range in this CO2 response emanates mainly from a lack of experimental data. Because we cannot bring, say, in case of forest, big trees into the lab to do all the manipulations and really mimic the true future. And within this context, the northern temperate forests, particularly deciduous mature forests, never had similar experiment to look at their CO2 uptake response under future climates. So the Birmingham Institute of Forest Research then established a 3-year carbon enrichment facility, the FACE facility, in a mature oak land. These oaks are about 170 years old. And we have the experimental setup, 3 ambient phase rings with the infrastructure, but no CO2, in 3 treatment rings, but where we raise the carbon dioxide concentrations by 150 ppm or units above the ambient from dusk to dawn, from dawn to dusk during the growing season. We also have non-infrastructure controls where we undertake essential background measurements to be able to separate the treatment effects from the controls. Baseline measurements were started in 2015-16, and then the fumigation started in 2017, and now we are halfway through, and it's going to continue up to 2026-2027. When the fumigation started, so some of the key questions that were raised and were to be addressed was how the elevated CO2 would influence the carbon budgets and flows across this ecosystems, what would happen to the nutrient cycles to be feeding the photosynthesis if there is limitation, assuming temperate forests are mainly limited by nitrogen availability, and in some cases phosphorus, and how the whole ecology of the woodlands from insects and bacteria and microbes all the way to the broader ecology would respond to that, and with specific impotence also on the resilience, diseases, and health of trees under future climates. I'm only focusing on carbon and nutrient cycle today because that's 15 minutes in total. But there are further presentations about the resilience and health of trees by Mojgan and few others, and many posters outside. So I will point you to those with time. So results above ground, starting from the above. So this figure shows the photosynthetic response in terms of under ECO2 work done by Anna Gardner, and it's across an increasing light intensity. The blue would show the control, and the orange color shows the elevated CO2 response. In the upper canopy, the response is more prominent. There is more CO2 and there is more light, so that response is more conspicuous. But if we average it all across the depth profile of this light intensity, over the first 3 years, on average, the oak-dominated systems took about 23% more CO2 than the control one. Linked to this above ground CO2 uptake, the first evidence, and now we just confirmed it this year, is evidence for enhanced tree growth under elevated CO2. Again, this work is mainly driven by Rich Norby, Neil Loader, Mayoral, and many, many more researchers all together. The initial differences between the treatment and control rings were adjusted for 2012 to 2015. So little difference, about 5% pre-treatment. And from 2017 onwards, there is year-to-year variations, but on average, 28% basal area increment increase now. This within the context of net primary productivity in the total budget, that talk will be given by Rich Norby later this afternoon. So watch for that to delve into the details that I have no knowledge of how it's being done, but Rich would be able to do that. Now, above ground responses, we collecting leaf litters by species in trees and all. This is only showing leaf litter for oak trees from 2015 to 2021. There is a consistent response, relatively more 14% increased oak litter fall under elevated CO2 from 2018 onwards. The average is 14 if we include all the 2017 where the response was not higher. So more litter fall going down, at least in case of oak. The interesting bit is this is half data that I received yesterday from Carolina. The concentrations of total nitrogen in the litter of the oak is about 12% less in the treatment compared to the control. So resorptions of the nitrogens, probably the tree is conserving the nitrogens so that it could feed the photosynthesis the next year. So that's one of the evidence. And if we move from above ground to below ground responses in terms of exudations of carbon and nitrogen by roots, here you could see these oak roots where we did these exudation studies in the middle of the pandemic. Mikhail Aria, who was the lead postdoc back then, August 2020, everyone was scared, but she was out accessing the roots and collecting the exudates. And the exudates were measured from that August 2020 all the way to June 2021. On average, the exudations of carbon increased by 4% based on root surface area. And look at the scale here, 0 to 5 microgram of carbon or nitrogen. But the exudation of nitrogen did not increase the total dissolved nitrogen, which resulted in a shift of the DOC to TDN ratios in the exudates from 4 to 8. So again, what we see above ground in conservations of nitrogen, we see a similar response below ground, conserving nitrogen potentially to be feeding into the trees. Here we see a below ground response in terms of soil respirations from 2017 to 2021, continuous measurements, more than 90,000 measurements so far. Initially, there was a stronger effect, 30, 40, 50%, and that's in the later years from 2020 onwards is on the decline. Overall, it's a 30% increase, but as we go, we see suppressions. This data, the first years is about to go to the Biogeosciences Journal. The manuscript is ready. The latter is still interrogated to reconfirm all the trends. Now, when the carbon allocations above ground and below ground increased, some of the carbon, particularly to the roots, that mobilize the activity of microbes. So that's one of the reasons trees do it, to mobilize microbial activity. And in that, we looked at the nitrogen as a first step. So we did measurements of the nitrogen cycling. And that cycling was particularly here, I'm showing ammonifications, the conversions of organic nitrogens to ammonium by these set of microbes. Initially, we started in lab 2018, 19, and 2020 using isotope tracing techniques. And we always saw some response, more stark in 2019 than in 2018 or 2020, but a bump in ammonification, significant only in 2019. We moved on to 2021 with Manora Mo as a PhD student, doing it seasonally now. Each season we do it in intake soil cores to see if the disturbed systems would show similar response. And we see so far a 25% increase in the field-based ammonification rates. These ammonifications, we do the same time the immobilizations of the nitrogen by the microbial body, because if their population grows, they need the nitrogen together with the carbon that is delivered by the roots, let's say. So the immobilizations also enhanced across the same experiment on average by 39%, this metabolic uptake. So bigger populations, bigger uptake, and that's the microbial turnover is faster. So probably eventually it should feed the trees for the required nitrogen. Now, we did the isotopic work. We wanted to double check, and that was mostly done in the lab, except in 2021. We wanted to check it in the field conditions using another technique. It's called the in situ net nitrogen mineralizations. It's also a part of disturbance on the side, but at least we started in 2022 with manure remove on a monthly basis. Net ammonifications is enhanced by about 36%, and so is net nitrifications by 45%, which is pretty much similar to gross mineralization we saw through the isotopic measurement. So it's a bump in the nitrogen cycle, internal cycling happening, and both the methods confirms the same trends. So pretty confident that the nitrogen cycle is upregulated. I'm not going to go into the details of the emissions of nitrous oxide driven by nitrifications in denitrifications microbial processes. But in 2018, 2019, and 2020, at the same time when we were doing gross mineralizations, we looked what is the relative contributions of nitrifications in denitrifications to the net emissions of nitrous oxide, which is the laughing gas in a dentist clinic, but a grim matter in the atmosphere. It's 300 times more potent than carbon dioxide in inducing global warming. So of course we have to account for the exchanges and fluxes of nitrous oxide as well other than carbon dioxide to do a true global warming potentials of the systems. These are potential rates based on lab incubations. So we're doing field measurements of in situ fluxes to reconfirm that what we see is happening on the field as well. Highly variable process, but on average, there is bump into emissions. Since we do these studies under additions of nitrate and ammonium as isotopic labels, so we also did another study in 2020 without the additions of the label, and we saw a similar response in terms of higher into emissions. Running out of time, so I think I better recap. So carbon in nitrogen cycle responses, higher uptake in the canopy, more litter fall with low nitrogen delivery into the ground, 40% bump in exudations, but change in DOC to TDN ratios. I did not show data for fine root biomass, but that's increased in the upper layers by that amount. There is increase in microbial biomass, carbon, nitrogen, and also phosphorus. So that's now coming on board. We are waiting for further measurements and data on the phosphorus dynamics. So that's also coming through. This carbon enhancement then resulted in an enhancement of the nitrogen mineralizations cross inlet and eventually into emissions. There's tens of studies underway covering. I did not have time to cover it all, but posters outside in subsequent talks. But overall, there is evidence for conservation of nitrogen by trees, upregulations of soil nitrogen cycle to sustain both microbial activity and probably potentially the CO2 fertilization effect. How long? Our forest is about 170 years old. Average age of English oak is about 150 to 150 years. Our halfway through, we see that there is potentials for these systems to be taking up carbon. But for how long? When will a new equilibrium be reached? A decade-long experiment is a first step and the most important one. But of course, Ram Oran, who ran the Duke base, he said maybe 10 years for the life of forest, 10 years is short. And it's a very expensive business. But based on science evidence, I think we have a conviction that maybe we may have an opportunity to go beyond a decade to assess and enable transient versus stable state shift responses. I will acknowledge all the collaborators engaged at BIFoR FACE and many more. And there are many, many researchers who contributed to this work, including our funding bodies. Thank you.